inviting me, dear Chairman. And I would have loved to be in China. I really like China. I've been there about 40 or 50 times, but now I'm sitting here in Amsterdam. But we'll do my best and discuss with you the potential role of nutrition in modulating the long-term consequences of early life stress. And I have prepared this talk together with some wonderful people like Hanna Juncker here, Britt van Keulen, Martijn Vinke, all from the Emma Children's Hospital and the Free University in the University of Amsterdam, Anniko Korosi from the University of Amsterdam as well, and Suzanne de Roy, who is also from Amsterdam UMC. And we have nothing to disclose in relationship to this work. So I'm going to talk about early life factors and how they determine the outcome and what we can do to improve the outcome. And it's good to know that both nutrition and stress have an effect on later neurodevelopmental disorders, on mood and on cognition of the offspring. And the effects are already there during pregnancy, where nutrition and stress play a role in altering the maternal biology, like metabolic and immune and inflammatory and endocrine function that all have an effect on the fetus in the womb. And if the fetus is born, then of course there is also an effect of infant feeding and later on of the diet of the child, of the physical activity, of postnatal stress exposure, maternal parenting behavior, or the postpartum stress, or the mood state of the mother. So all these factors have an effect on neurodevelopmental disorders, mood and cognition of the child in later life. What do we know? Because this is just a cartoon showing what is happening. What do we really know of these mechanisms? Well, we have some observational data. These are observational data from Chile, more than 50 years ago, where they looked at brain weight and brain protein, DNA, and RNA content of infants that died in accidents or of infants that died after severe malnutrition and probably also severe stress. And here on the left upper panel, you see the brain weight, where you have the dots that are the normal Chilean children, and the triangles here are the ones that had who died from severe malnutrition during the first year of life. And you can easily see that the weight is reduced in those infants that suffered severe malnutrition. But not only the brain weight, also the brain protein here, or the brain RNA, or the brain DNA. And actually, if you look at the pattern, you can see it's almost identical of those who died from severe malnutrition, suggesting that the reduction in DNA was also accompanied by a reduction in weight, which suggests that there is just reduced cellularity in those brains of those infants. This is another study from 20 years or so ago, from brain autopsy in Mexico, from infants less than 2 years of age, where they counted the dendritic spines from cortical neurons. And here you can see 7 normal children, and here 13 data from 13 infants who died of severe malnutrition. And you can see that the average length of the apical dendrites was reduced in those who were suffering from severe malnutrition, as was the number of the average numbers of spines, both indicating structural changes in the brains of malnourished children. Then, as we are in China now, or we should have been in China now, I compared 2 famines, one from my own country. The Dutch famine was what that took place in 1945 during the end of the Second World War, where individuals were exposed to a very severe protein energy malnutrition diet with only 400 kcals per day, and also the Chinese famine, which was around 1960, so 80 years ago now. When one looked for an association between the famine that occurred either in early childhood or during gestation, and looking at cognitive function at 19 years of the adolescents or the young adults, there was no effect. And also, when you look grossly to cognitive function, there was at 60 years also no effect. But in the Dutch family, when one looked more closely to those who were grown up to 60 years, they found a lower selective attention, which is indicative of the prefrontal function and indicative of early cognitive aging. And exactly that was found in the offspring of the ones who were born or raised during the Chinese famine. Here you see the results from China. Here are a group of now around 50, 55, around 60 years of age of non-exposed people. Here in the black bars, those are at present, at the time of the study, they had a high nutrient consumption pattern and a low nutrient consumption pattern in the open bars. And on the y-axis, one shows the prevalence of global cognitive decline. And you can easily see that when they were non-exposed, it's around 10 to 15%. Also, when they were exposed during the late childhood, it was also around 10%. But during fetal life or early childhood exposure or mid-childhood exposure, you can see it increases up to around 20%. So a clear effect of cognitive decline when you were exposed to the famine and the stress accompanying the famine in early childhood, early life. So adversity early in life, like stress or malnutrition or poor growth, is associated with a negative impact on brain development, on behavior, but also on cognition. What are the possible mechanisms? Well, then it's important to understand how the brain develops in early life. You have in the first trimester neuronal proliferation, the second trimester neuronal migration starts, synaptogenesis starts, apoptosis starts, and myelinization starts at the third trimester and continues, of course, during the early few years of life. Importantly, total brain volume triples during the third trimester. The number of neurons peaks at 28 weeks of gestation, and at 34 weeks around here, 40,000 synapses are formed every second. So it's important to identify at what time an insult is taking place because it can have differential effects on the brain. So perinatal malnutrition, depending on the timing of the insult, can have an effect on cell proliferation, on migration, on the fate. It can have an effect on the function of the neurons, on neurochemistry, redox status, on connectivity. It can have an effect on glial function, on the gliosis, on the myelinization, microglial dysfunction. And all these factors can have differential effects on physical development, on neurological deficits, on impaired learning and memory, and on socio-emotional dysfunction. So what are those mechanisms? How can it exert its effect? Well, by far the most important modulation factor is the hypothalamic pituitary adrenal axis, the HPA axis, but also oxidative stress, epigenetic factors, inflammation, and just the regular deficits of essential nutrients may play a very important role. Just to go back to your textbook from your studies, the hypothalamic pituitary adrenal axis and stress. How does that work? Well, you have the hypothalamus that releases CRAs, the corticotropin releasing hormones that stimulates the anterior pituitary gland that on its turn produces adrenocorticotropic hormone, ACTH, that stimulates the adrenal cortex that on its turn produces cortisol and corticosterones. And those cortisol has a negative feedback on hypothalamuses, reducing the CRH release, or has also a negative feedback on the anterior pituitary, and thus regulation of the ACTH levels. So if you look at animal experiments, if you stress them, so the pregnant mothers of rodents, here mice, and you look at the male offspring in this study of Abe in 2007, they found that when those mothers are, during pregnancy, are stressed, the offspring at about a week of age have higher basal corticosteroid. So this HPA axis is being stimulated. And then when you're looking at, for instance, mood states of those offspring of those young mice, they did a test which is called the forced swim test, which is a sign of depression of the mood. What they do is they throw those mice into the water and they look how long it takes before they start swimming. And if they're not stressed, the control, it takes about 30 seconds. So they're lying in the water and say, ooh, now we need to swim, otherwise I drown, and that takes about half a minute. If they're stressed, if the mothers are stressed, and that has an effect on the offspring, it takes them more than a minute before they start swimming. Another mechanism is through oxidative stress. One usually thinks of oxidative stress that it has a negative effect. It's also in those creams that prevent aging of your skin. So it has a negative annotation, oxidative stress. But you really need some oxidative stress because it's important for proliferation and differentiation of cells. Only when it's very high, it induces apoptosis and necrosis. So low amounts of oxidative stress is good. High amounts of oxidative stress is not good. One way to induce high levels of oxidative stress is to add fluoride to the drinking water. So here, this is a test again in rodents where they provided the dams during gestation and during lactation with 5 or 10 milligrams per liter of fluoride in their drinking water. And then they tested the hippocampus by what is called the step-down inhibitory avoidance test. It's just you put a rodent here, a mice on the plateau, and of course the mice starts wandering around because he's curious. But on the floor, you have a device that gives a shock to the mice whenever they touch the floor. So if they jump off, they get shocked, and of course they jump back. So when you look at normal mice that are not being exposed to fluoride, you see that if you do that for the first time, it takes them about 1 second because they're so curious, they jump off the plateau and then they jump back. But within 1 second, they jump off the plateau. But after 90 minutes, after they've done this the first time, they don't dare to jump off the plateau at the beginning. It takes them about a minute, and then they become so curious that they jump off anyway. But then after 1 day, they remember, and it takes them even longer before they jump off. So then they're smart and they think for 1.5 minutes, but then still they jump off. When they are exposed to fluoride, the mothers of those infants, and they have high levels of oxidative stress during pregnancy, but also during the lactation period, you see they don't remember that well. So instead of remembering for 1 minute that they shouldn't jump off, they only can recall that for 30 seconds, and at the high dose even only for 10 seconds. And 1 day later, 90 seconds, but only the ones that are exposed to high levels of oxidative stress, they can only remember it for 40 or 40 seconds. There's a difference between males and females in that reaction. So that is about the mechanism of oxidative stress that affects the hippocampus function. Then epigenetics. Epigenetics, you all know, probably it has nothing to do with the content of your DNA, but more of the expression of your DNA, how much proteins are being derived from your DNA. And this is an interesting experiment where you have a mother that is not taking care well of their pups, so with low licking and grooming, comparing to a very nice mother that is highly licking their offspring and grooming with them. that has an effect on the glucocorticoid receptor expression. Low licking and grooming, low expression. And as a resultant, you have high corticosteroid levels in the offspring, high anxiety, and on their turn, the female offspring becomes a low licking and grooming mother. Whereas the high licking and grooming mother has a high glucocorticoid receptor expression in the offspring, so therefore low cortisol levels, low anxiety, and on their turn, they become very nice mothers. Then we go to humans again. This is about the mechanism on inflammation. Here they have looking at associations between the exposure of pro-inflammatory cytokines in utero, in humans, and then looking at functional MRIs made at about age of 45. And again, they find a difference in connectivity and in activity of different regions in the brain, depending on low inflammatory cytokine levels or high inflammatory cytokines levels. And there was also an effect on a differential effect on gender. So for instance, high IL-6 in utero decreases the hippocampus activity, but only in females and not in males. So it's all interrelated in this study as well. The HPA axis is a critical junction for immune factors, for steroid hormones, and as a result in the regulation of stress responses. Then the last mechanism that I like to discuss with you is on nutritional deficiencies. And here, for example, is the example given on iron deficiency or anemia. It has an effect on hippocampus IGF signaling. Iron deficiency through the IGF pathway reduces in reduced proliferation or cell growth within the hippocampus. And that accounts for many of the early nutrient deficiencies on copper, on iodine, or thyroid hormone deficiency, or on protein energy malnutrition. They all result in hypomyelinization, in abnormal cognitive function. And for instance, copper deficiency induces again iron deficiency, and they both induce a hypothyroid state in the brain. So they're all interrelated again. So are we able to prevent long-term neurological abnormalities associated with perinatal adversity? Yes, I do think we can. We can help in reducing the amount of stress exposed to mothers or infants. For instance, like programs like family integrated care, where we know it reduces stress levels in the mothers, which we hope then will have positive effects on the long term of their infants. One could try and interfere with genetic factors. And I will show you an example later on. One can try to improve the defense mechanisms against increased levels of oxidative stress, like cystathione supplementation or providing human milk instead of cow's milk formula. One can also, of course, be at the source of oxidative stress, like oxygen, and provide less oxygen to infants at vulnerable states. One can reduce inflammation by preventing infections, also those low grade, like coagulase negative staphylococci infections, which we all know are not, but the infants are not dying from it, but it induces the inflammation of those preterm infants. And of course, prevent nutritional deficiencies. So one should know the requirements and provide the nutrition that is needed to those infants. Just some examples. This is on methyl donors prevention. So here we did a study where in rodents again, we provided from postnatal day 2 to 9, when the mothers were lactating to the small mice. We gave them a micronutrient supplemented diet with choline, betaine, folic acid, vitamin B6, B12, methionine, zinc, a 1-chem diet, what we called. cm. And here you see the results. At P9, providing the mother with this diet did have an effect on cortisol levels, because if you put stress to the mothers by taking away nesting material, the offspring had higher cortisol levels, but providing early life stress with this diet, cortisol levels were normal again. Then looking at more cognitive outcome, this is at 4 months. In the standard group, early life stress, less object recognition, but when those mice were provided, or the mothers were provided with the specific micronutrient diet, this effect was abolished. There was no effect, interesting, on DNA methylation in the brains of those offspring, on glucocorticoid receptor expression, neurogenesis, or hippocampal volume. Another example on meat requirements, the effects of omega-3 PUFA availability, so polyunsaturated fatty acids. Early stress exposure, again, during the lactation, it changes peripheral and central fatty acid profiles in mouse. Then by increasing the omega-3 ratio to omega-6 ratio, so with a low omega-6 to a higher omega-3 ratio diet, it prevented early stress-induced cognitive impairments like object recognition. In the unsupplemented diet or the regular diet, a clear effect of the ones that were exposed to early life stress, whereas the diet prevented that effect. Morris-Weiss maze probe trial, no effect after anymore. Object location, again, much better after providing a diet with a higher omega-3 PUFA availability. So in conclusion, the effects of perinatal psychosocial stress and malnutrition, both during pregnancy, but also during lactation, so also on probably the effect on the diet, on the milk of the mammal. Those effects are context dependent. They are simultaneously influencing one on each other at various physiological and behavioral levels, and should not be considered in isolation. The identification of the interplay of perinatal stress and nutritional factors may provide us with new clinical intervention pathway, and thereby improving infant and maternal health with long-term health benefits for the child. So we require carefully designed animal and human studies to identify the primary nutritional factors, so we can target those effects the best to ameliorate the adverse effects of early life stress on child neurodevelopment. Thank you very much.